Hello, we're glad you've joined us for this live webinar, Mate-Pair Next Generation Sequencing as a Powerful Clinical Tool for the Characterization of Cancer. I am Christina Mahalik of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. To learn more, visit labroots.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left of the screen. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I now present today's speaker, David Smith, PhD, professor and consultant at Mayo Clinic. David Smith received his BS in mathematics and molecular biology from the University of Wisconsin in Madison. He then received his PhD for the Department of Biochemistry, also at the University of Wisconsin in Madison. After several postdoctoral positions, he got his first academic appointment at Wayne State University. In 1996, he joined the Mayo Clinic in the Department of Laboratory Medicine and Pathology as professor and consultant. He was also appointed as the head of the Cancer Genetics Program for the Mayo Clinic Cancer Center. His research focuses on the role of the common fragile styes play in cancer development. His laboratory also works on the various roles that the human papillomavirus plays in development of different cancers. Dr. Smith is also the chairman of the Technology Assessment Group for the Mayo Clinic Center for Individualized Medicine. The responsibility of this group is to evaluate new technologies for their potential impact on research and its clinical translation. His laboratory utilizes next-generation sequencing to characterize the molecular alterations that occur during the development of cancer. They also have been using the powerful technique of mate-pair next-generation sequencing, MP-Seq, to characterize the physical status of HPV in different HPV-driven cancers. Dr. Smith will now begin his presentation.
Thank you, Christina. It's a pleasure to be here today. Again, I'm going to be talking about mate-pair next-generation sequencing and why I believe this could be a very powerful clinical tool for the management of cancer patients. All of this starts with next-generation sequencing, which are technologies which are based upon massively parallel sequencing. One runs hundreds of thousands to billions of simultaneous sequencing reactions. And what differs are the various technologies that are utilized to perform the actual sequencing. The very first viable next-generation sequencing was developed by a company called 454 Life Sciences, and this is a picture of the actual sequencing box, where the sequencing and the chemistry is done in the white box, and the top little screen with the TV is where you can monitor everything that is occurring. The key of all types of first-generation, next-generation sequencing was to figure a way to amplify individual DNA molecules to a sufficient number of molecules so that one can get a sufficient signal for the sequencing of that individual molecule. And the 454 platform utilizes something which is known as emulsion PCR to produce that clonal amplification. The key features of this emulsion PCR is the fact that you are making individual little reaction vesicles, which are tiny little water droplets in a liquid interface. And what one does is in these tiny little droplets that only contain maybe 50 picoliters of water, you are actually placing several things. The first is a bead, which is shown as the little bead on the top right-hand corner. The second is an individual DNA fragment. And the third is the necessary reagents to amplify that DNA fragment. And the fourth are oligonucleotides on the surface of the bead. And what one does is one does a very dilute concentration of these beads, individual DNA molecules, so that you're not amplifying multiple DNA molecules in an individual bead. And at the end of emulsion PCR, where in that bead where you had a single DNA molecule, you were able to amplify that DNA molecule up to 1 million copies. And the second thing is so if one goes through the entire process, one takes your DNA source. That could be an entire genome or a portion of the genome. And those DNA fragments the DNA is cut into fragments, and the fragments have primers, we could call them A and B, that are placed on the 5' and 3' end of those fragments, and then the individual fragments at a very dilute concentration are placed into the emulsion PCR to amplify an individual fragment. So where there was once an original fragment, there's up to 1 million. One has a very dilute concentration of that so that there's no more than one DNA fragment amplified per bead. Since the majority of the beads have no DNA fragments, they don't amplify any DNA, but one can purify the fragments with amplified DNA from the ones that do not on some type of density gradient. Once one has taken that and has basically broken the emulsions, you can take the individual beads and place it into the sequencing surface, which is actually a picotiter plate. It's called a picotiter plate because it holds picoliter volumes, and it's actually produced from a telecommunication cable, which basically has 1.5 million very thin fibers whose diameter is 30 microns. The diameter of the beads on this platform is 28 microns, and then when one places that in, and shown on the extreme right is the bead with the necessary reagents for the PCR, not for the PCR, for the sequencing, and it utilizes pyrosequencing, which can detect the incorporation of a nucleotide by the release of light. So then one places that into the machine, then has 4 different containers that contain individual nucleotides. These nucleotides are naked. They have nothing on them. They're not blocked in any way. One floods this with A and asks which of the wells produces a signal through pyrosequencing, which is a light reaction. Then one floods it with T, G, and C, and then one continues this as one is incorporating bases. On the next slide, one can see the same picture again and the actual output from the sequence of a single bead. So you can see that the very first base incorporated was a T. When one cycles all 4 bases, T, C, G, and A, the T was incorporated. Then one does the cycle again for the next base, and you can see it's a C. However, you can see that some of the peaks are higher. You see where you have 2 A's being incorporated. Because the bases are not blocked, one incorporates both A's if there are 2 T's that require an A to be incorporated. Further over, you can see the incorporation of 4 G's. So this is the output from a single bead from the first generation of this machine, and you have 191 base pair read. Very quickly, they were able to get the read length even longer. Now, the strengths and weaknesses of this platform was that it was the birth of next-generation sequencing. It was capable of doing long sequence read length that was not much shorter than Sanger, and this was a real strength. And it transformed sequencing. The predecessor to the 454 platform was Sanger sequencing, which was actually utilized for sequencing the human genome at a cost of over $200 million for the sequencing. And yet in 1998, on the developed 454 platform, the sequence of Jim Watson, his whole genome was sequenced for a cost of only $1 million, which is a transformation in terms of the total cost for genome sequencing. But there are notable weaknesses to this platform. One was a limited room for improvement. The first generation machine was capable of 20 megabases of sequence. With improvements, they were able to improve that to 500 megabases per run, and this is how the Watson genome was sequenced, but there was no room for further improvement. They couldn't get read lengths greater, and you couldn't have a larger number of beads because that's all the microtiter plate could hold. The second real weakness was the emulsion PCR, which was an extremely messy technique and difficult to do. And the third, already mentioned, is this whole homopolymer issue. If you have a long string of nucleotides, they are all incorporated because the bases are not blocked. If one wants to utilize this in a clinical sequencing setting, there are a number of positions in the human and other genomes as well that have long runs of homopolymers, and it is very difficult to discern the difference between, for example, 10 nucleotides being incorporated and 11. Hence, there's going to be sequence inaccuracy at those positions. The second viable sequencing platform to be developed was, and this is from the company called Illumina, which was actually purchased from a company called Solexa. Their first generation machine was the Illumina Genome Analyzer, and this on the bottom is the actual reference from Nature of this sequencing platform. The Illumina Genome Analyzer had a number of notable advantages and improvements over the 454. The first was an improvement as a way to amplify DNA. Rather than have DNA fragments that were amplified in emulsion, one places on the surface of something which is known as a flow cell the same types of nucleotides. One could call them A and B. The DNA fragments are then produced the same way as on the 454, and one flows the DNA fragments across the flow cell, and what happens is that DNA fragment, the primer at one end is complementary to one of the primers. There's a high density of those primers on the flow surface to them, and then the other end is complementary to the orange, which is now on the surface. And what can happen is that DNA fragment can bend over, and now you've produced basically a DNA fragment that is now bridged, and hence this is known as bridge amplification. In the presence of polymerase, this DNA fragment can be copied so that you can basically now have two copies. If you denature that, now you have two copies at that position, and one can repeat this process, and by the time you're finished, where there was an individual DNA fragment, now you have 30,000 DNA fragments, which is sufficient for this platform for the detection of signal. This flow cell can then be taken from the machine where it is produced and placed into the sequencing machine. The top right actually shows the first generation of how these clusters were actually done and how bridge amplification was done. And now on the surface of the flow cell, where you had individual DNA molecules, you now have these clusters, but across the surface of the flow cell, there were millions of these clusters. This flow cell is then taken and placed into the sequencing machine, and in the sequencing machine, you can then basically do sequencing. And a second advantage of this platform is instead of having unblocked nucleotides, the nucleotides themselves are fluorescently labeled, G one color, C another, T another, and A another, and then one can do all four of the incorporations simultaneously rather than flowing one at a time. One adds the nucleotides, and in the presence of a primer for sequencing, one will incorporate an available nucleotide at each position, and you can see on the middle there's one cluster that gets a G, one gets a T, one gets a C, and one gets an A. One takes a picture of that, and the top right is the picture of the flow cell and the incorporations of each position. After that is done, only a single base can be done because the fluorescence blocks the nucleotide from incorporating another base. But in the presence of something to remove the fluorescent nucleotide, now that base that is capable of adding a second base, and you can repeat the process. And then you take a picture there. That is shown in the top right to the second panel. Then you see the third, the fourth, and the fifth. And if one takes a look, for example, at the sequence of the cluster on the left, the first base incorporated is a G, then a C, then a T, then a G, then an A. That G, C, T, G, A is the sequence of that single cluster, and one does this simultaneously for hundreds of thousands of individual fragments. However, the original Illumina sequencing machine, 3 years after it was developed, the new machine came out, and this was the HiSeq 2000. Once they realized that the flow cell has two processes, a chemistry process and a scanning process, they realized that they should probably have two flow cells, one that does the chemistry, the second that does the scanning, and the HiSeq 2000 was capable of running two slides, two flow cells rather than a single one. And the improvements on the HiSeq The first thing is when clusters were originally made, the clusters actually were on the top and the bottom of the slide, but the original sequencer only read and had a focal plane at the top, but now they were able to have focal planes at the top and the bottom, which doubled the number of possible sequences. There was a faster scanning of slides, it was capable of longer read lengths, and greater number of clusters per slide, but very quickly this obsoleted the genome analyzer with less than a 3-year life cycle, and this is a disadvantage, especially when each individual machine costs just shy of $700,000. Now, if you look to the bottom right, one sees the undefined features when you have random spacing, when you use these flow cells and you just randomly amplify clusters. This worked quite well, and increased output on this machine went up at least 100-fold, but eventually you reach a point where it becomes very difficult to discern one amplified cluster from another. And what happened is the next advance was known as the pattern flow cell, and this is shown on the bottom left. Rather than just have a flat slide surface and random amplifications, there are amplifications that are occurring in these pattern flow cells. Each individual little dimple is an individual place where a single molecule can be amplified, and when one sees the picture, and this is shown in the bottom middle, you get much more ordered spacing, which makes it much easier to tell one sequence from another and gives you the capability of producing a higher density. Higher density means there are more sequences across the flow cell, which means greater sequence output. And then what one has is an entire space of different sequencers from Illumina, and this is actually an old slide, which actually, because there's a new platform that has come out, but at the bottom left is the lower output machine, which is the MySeq. This machine produces 15 gigabases of sequence, 25 million reads, by producing 300 base pair reads at the ends of fragments. One should realize that the original sequencer from Illumina was capable of only a single gigabase of output. And the read length on the original machine was only 30 base pairs, which made it very difficult to assign where those fragments were in the genome. There was a second platform that had a greater output of 120 gigabases, the HiSeq 2500, which was a 1 terabase of sequence output, and then the first viable machine that was utilizing the pattern flow cells was the HiSeq X10, which produced 1.8 terabases of sequence, 6 billion sequences on basically 2 times 150 base pair reads at the ends of the fragments. And these machines were very powerful, but you had to purchase 10 of these machines at a time at a cost of $10 million. However, the strengths and weaknesses of this platform are really important. One is currently the lowest cost per base for sequencing of any sequencing platform, and it is the only viable platform for whole genome sequencing. However, I should mention that BGI, the Beijing Genome Institute, does have a sequencing platform that, if made commercially available, could be a second platform that is a viable platform for whole genome sequencing. Currently, Illumina completely controls the sequencing space. They have block nucleotides, which means there is no homer-polymer problem. But there are weaknesses to this platform. These high-throughput machines need sufficient sample, or you're wasting space. If you turn the machine on and turn the key, you are utilizing the full cost of the reagents, which is about $25,000. If you do not have enough sequences on that machine, the net cost per sequencing goes up. And there's a trouble with small amounts of input nucleic acids. If you have a limiting amount of nucleic acids, for example, from clinical samples, it may be difficult to get signal from those limiting nucleic acids. The customer service is a potential problem because with the absence of sufficient competition, there's really no motivation for Illumina to have ultimate customer service, because the customers really don't have a choice. If you want to do whole genome sequencing, this is the platform you have to work on. The second one already mentioned is each time you have a single platform, the instrument cycle time is relatively short, which means very quickly a new machine comes out which obsoletes all the machines that you have. And one other question, one of the goals of genome sequencing was to make it affordable, so affordable that it could be routinely used, and the thought was that that should be a $1,000 genome, but I suggest, what about the $100 genome? And could that be achieved on this platform? There's really no motivation for Illumina to push to get lower sequencing costs if they do not have competition. However, recently Illumina announced a new platform, and this is the NovaSeq 6000, which is the newest generation machine. There are actually 2 machines, the NovaSeq 5000 and the 6000. The advantage on these machines is shown on the next slide. These machines actually have flexible flow cells that one can run. Currently, or soon to be available, is the 0.5 terabase flow cell and the 1 terabase flow cell, and this gives you greater output than you could have on the previous platform. And since the NovaSeq 6000 actually runs 2 flow cells, with the 1 terabase flow cell, you run this machine, you produce 2 terabases of sequence. Soon to be released, presumably the end of 2017, early 2018, are 2 higher throughput of flow cells, one with 2 terabases of sequencing, one with 3 terabases of sequencing. With the 3 terabase flow cell and running the NovaSeq 6000, when you turn this machine on, you are producing 6 terabases of sequence output, which is 4 times greater than the HiSeq X10. On the HiSeq X10, the cost to sequence a genome, just the sequencing, was less than $900. With this platform and 4 times the output, one reduces that even greater, and one starts to see an approach towards even cheaper genome sequencing. Now, there is another platform, and this is a platform that is very similar to the 454, and it's not surprising because it was developed by the same person who was responsible for developing the 454 sequencing platform, Jonathan Rothberg. And very similar to the 454, this has the same type of amplification, emulsion PCR. The bases are unblocked, so it has the same issue that the homopolymers are a problem. And while this platform came out after Illumina, it's been trying to play catch up to Illumina, but it's not working. The Illumina platform has much greater output and has certain advantages over the Ion Torrent platform, and anything the Ion Torrent can do, Illumina can also do. Hence, this represents at best an incremental technology, and still Illumina completely controls the sequencing space. However, there are some strengths to the Ion Torrent platform. The first is turnaround time. This is a faster sequencer to produce sequence. And it has a lower input amount of DNA, which is necessary for selection protocols. So if one is doing sequencing of a small gene panel, for example, one can use smaller amounts of input DNA. And as a result of this, the National Cancer Institute chose the sequencing platform, the PGM, which is the personal genome sequencer, from them over the Illumina MiSeq because of this one fact. And indeed, there are a number of clinical labs that are adopting this platform because they can get results from a higher proportion of samples, especially samples that have small amounts of material. There's also viable solutions to simplify emulsion PCR because, as mentioned, emulsion PCR first generation was much messier, more difficult to do than bridge amplification on the Ion Torrent platform. The Ion Chef is a viable solution that makes it easier to do without having to work with the messiness of your hands of the lipids. And it has flexible output because on the Ion Torrent platform, it is capable of running multiple different size things depending on what you want to sequence, and this platform has the thought and the strength that there could be further improvement. And indeed, in the number of years that the Ion Torrent platform has come out, it has increased its sequence output dramatically. It just hasn't been able to get even close to the Illumina platform. Hence, the Ion Torrent platform is not a viable solution for genome sequencing now, and that raises the question, will it ever be available for that? Now, there is a next generation machine, and this is the first of that. This is the Pac Bio RS. Pac Bio, Pacific Biosciences, has now an improved machine over this one. This is just an older picture. This is the first single molecule sequencer. Rather than amplifying DNA fragments to a large number of copies and then getting signal from those multiple copies, this machine sequences single molecules, and this has certain strengths that I'll mention in a minute. This single molecule sequencing does real-time sequencing by tethering a DNA polymerase at the bottom of a tiny little well, which is known as a zero-mode waveguide. This has fluorescently colored nucleotides, the 4 bases, A, C, G, and T, and as each base is incorporated through that polymerase, fluorescence is released, and in the zero-mode waveguide, which confines the wavelength of the signal that's being released, one can see the output of an A and a T from 2 sequences, and this thing is a very fast sequencer because polymerase moves much quicker than the slower chemistries of the 454 of the Illumina platform. However, there's notable weaknesses to this platform. The accuracy is only 85%. This machine is so quick, you do not see every base that's incorporated. A second serious weakness is the output is low, and there's been relatively limited improvements since becoming available. Now, it has improved at least 1 to 2 orders of magnitude, but unfortunately, this is by far the most expensive platform cost per base for sequencing. Now, because of the inaccuracy of sequencing, there is the use of smart bells. These are the placing of basically bells on the end of a DNA fragment so that DNA is actually sequenced around and around so that one interrogates the bases more than a single time. Because the inaccuracies are totally independent of base, by sequencing the same base multiple times, one can achieve the same sequence accuracy as the other platforms, but this just decreases the output further and also decreases the read length. The strengths of the Pac Bio, however, and these are important, is much longer read lengths. With the newest generation of Pacific Biosciences machines, average read lengths are 10,000 to 20,000 base pairs. This is thus a viable instrument for completing genome sequencing because one can use this to scaffold together short sequence reads from the other platforms, and it's also a very viable thing for reading the sequence of an entire transcript. Some RNA transcripts are considerably longer than the sequence output capable on the 454 Illumina or the Ion Torrent platform. Hence, this can be used to sequence an entire transcript and enables you to determine exactly what the transcript is and the difference between different transcript isoforms. Since they're long reads, that enables you to get through difficult regions like the HLA, Fragile X, etc. And Pac Bio used to be in partnership with Roche developing clinical assays, but earlier in 2016, Roche dropped the Pac Bio, which was a severe blow to Pac Bio as a viable sequencing platform and having any clinical utility. Now, there is another viable single molecule sequencer, and this is from Oxford Nanopore. It utilizes basically a variety of different pores, which has a very small footprint, some slightly larger than a USB memory card, and like the Pac Bio, this is a very long read sequencer. It uses fluorescent detection of bases using different strategies depending on which pore one is utilizing. And this is the actual footprint of the Oxford Nanopore sequencer. And this is a very important thing because it's a portable sequencing machine. They utilize biological nanopores, which are protein pores that are naturally found in cell membranes. And with this, and this is a picture of one type of pore. This is a solid-state nanopore. And on this one, you either can do the solid state or you can use the biological nanopores. But one of the things that's illustrated here is that not a single base at a time goes through the pore. Actually, on the current Oxford Nanopore, there are 6 bases in the pore at any one time, and thus it's very difficult to know definitively the sequence of that, and this has sequence accuracy very similar to the Pac Bio. So the strengths is a small footprint. It's ideal as a portable sequencer, hence it's useful in the field or in space. And indeed, the Oxford Nanopore has been placed into space, and there's been some sequencing performed in space because you don't need a very heavy machine. It can be carried, and there's been dramatic improvements since this has first been made available. The weakness is the accuracy is terrible, very similar to the Pac Bio, and the output still has a very long way to go. Can they ever be viable for whole genome sequencing? And that is in question that I do not know currently, but it does not look like in the next couple of years, and possibly the next 5 years, that this will be a machine where whole genome sequencing can be done, similar to the Pac Bio machine. So there are other next-generation sequencing platforms. I'm not doing this to mention all of them, but for example, NuBio does sequencing by hybridization, which is potentially very promising for small gene panels. And a number of years ago, I believe 2 years ago, this was purchased by Bio-Rad. QIAGEN has developed an integrated end-to-end next-generation sequencing solution. They have single-sided sequencing by synthesis, but they also have everything from sample preparation, library preparation, sequencing on their sequencing machine, and they purchased an analysis company, Ingenuity. So basically, it's an integrated solution for everything. There's also DNA Electronics. DNA Electronics owns the Ion Torrent patents, and they are an easy platform for small genome panels, and they're currently developing a sepsis test. Genia is utilizing a biological nanopore and planar electronic sensors, and indeed there are many other platforms that are in development. Now, what are the impediments to a new next-generation sequencing technology? One of the serious impediments is that the Illumina Platform, which has now been in circulation for 10 years, has made dramatic improvements. Hence, any new platform that comes along that's just an incremental improvement will not be competitive. So what is truly needed is a real game changer. For example, a platform that can do whole genome sequencing for $100 with a long-read technology that is accurate. Currently, this is merely a pipe dream. It would be really nice if this was available. Or a very simple platform for small gene panels that puts together DNA isolation, selection, and sequencing, and is quick, accurate, and very cheap. And this is the thought that some of the things, for example, that I mentioned on the previous slide, one of these could possibly be this. However, again, if one of these platforms comes out and is merely incremental to what is available for Illumina, this is not going to be viable. Once this type of sequencing, for example, on the Illumina platform, is in clinical practice, they are very loath to switch platforms unless the platform is such a difference, such a game changer. So again, once something has been adopted, it is very difficult to change. Now, with the sequencing platforms, and again, I'm going to discuss mainly Illumina and Ion Torrent, there are different types of next-generation sequencing. You can do whole genome sequencing, although currently this is only viable on the Illumina platform and potentially viable on the BGI, the Beijing Genome Institute, BGI-Seq 500 platform. Hence, once that's commercially available, it may offer some competition. One can also do small gene panels, sequencing of a smaller thing than the genome, maybe a certain number of genes involved in some clinical disease. One can do something larger than that, which is known as whole exome sequencing, and this is the sequencing of the coding portion of the genome. In contrast to the 6 gigabase genome, the exome sequencing is only 38.5 megabases, which is considerably less. In addition, one can sequence RNA. This is known as RNA-seq. On the Illumina platform, RNA needs to be, and also the Ion Torrent platform, RNA needs to be converted into cDNA, and that cDNA can be sequenced. One can also utilize these platforms for the sequencing of methylation, specifically the 5-methylcytosine methylation, and one can thus determine the sequence of the epigenome. And this can also be utilized for something which is known as the liquid biopsy, which is getting a lot of press and a lot of interest. Now, whole genome sequencing is an extremely powerful tool to examine changes across the genome. It detects point mutations, copy number variations, translocations, and deletions. But there are problems with that. On the existing platform, this is before the NovaSeq is made commercially available, the total cost of less than $1,000 is just the cost for sequencing. One still has to produce the libraries, and that's maybe $100 or less. But what about the analysis? How does one assemble that sequence together? And when one does, what do alterations outside the coding portion of the genome even mean? And then there's the important issue, for certainly in the cancer realm, what if one finds an alteration? How many of those alterations are actionable? And this is definitely the case with whole genome sequencing for cancer, but it may also be true in other settings. And then there's the issue about turnaround time. I should mention that there is, and actually now, in the Guinness Book of World Record, the world record for sequencing and doing whole genome sequencing and analyzing and bringing it back to the patient has been brought down to a cost of 24 hours. And this is a big deal, and this is now being utilized clinically in neonatal intensive care units. But what will it take the whole genome sequencing to take off? The first thing is it depends on which patients are getting whole genome sequencing. Are we talking cancer care and treatment? Or are we talking about everybody getting genome sequencing? And indeed, if it's going to take off, it's got to take off with the total cost coming down. Library prep has to be considerably less than it is. The sequencing has to be at or around $100, and indeed this is possible with the NovaSeq. With the 3 terabase flow cells in NovaSeq, the cost for a whole genome sequencing could be $200 to $300, but then analysis and interpretation has to cost considerably less. I'm suggesting cost of maybe $250, and figuring some way so the storage of the sequence data is $100 to $200. If one adds all of this up, one sees that even though the sequencing could be $100, the total cost for whole genome sequencing is considerably greater, but this would be the instance where you would have whole genome sequencing at a cost of less than $1,000. And indeed, if one sees the increase of sequence output that's occurred over the last 10 years, this is not an endpoint. there is the capability of it costing considerably less. My suggestion is that when whole cost for genome sequencing, analysis, and storage is $100, this will be the total transformation, and it will be very difficult to argue why whole genome sequencing is not utilized in all settings, at birth, at any disease, for characterizing any types of alterations. And my suggestion to sequencing companies is to recognize and realize this, and if they want sequencing to be done at orders of magnitude over what it's done now, that is the endpoint they need to reach. Now, there are problems with whole genome sequencing currently, as I mentioned. The total cost is much greater than just the sequencing. But there is a viable technology that exists today that utilizes the strength of the Illumina sequencing platform, and this is known as MAPE next generation sequencing, which is abbreviated as MP-Seq. This is a powerful technology to analyze genomes. On the Illumina platform, one breaks the genomes into 3 to 5 kilobase pieces, biotin labeling the ends, and then circularizing those fragments. And then if one breaks the circularized fragments into small pieces and just captures the 2 ends, you can basically do paired-end sequencing of the 2 ends, and basically then do libraries to do paired-end sequencing. And this is illustrated on this slide. DNA is isolated and fragmented. One size selects the fragments to a specific size. On the Illumina platform, they utilize 5 kilobase fragments. They put biotin at the end. One circularizes the molecules. This is a low efficiency reaction, but the circularized molecules are immune to endonucleases, which would digest away any of the linear fragments, so one is left just with circularized fragments. If one circularized DNA, if one fragments that, the fragment that has the 2 biotin ends can be purified from all the other fragments with streptavidin-coated magnetic beads. One selects those fragments, and they're suitable substrate for paired-end next-generation sequencing. One does the sequencing of those fragments on a next-generation sequencer and aligns those sequences to the reference genome. For example, if this is done on the human, to the human genome. One generates this 1 to 200 base pairs of sequencing from the ends of these mate pair fragments, but these fragments were originally 5 kilobases apart. So that even though you've done only this 200 bases of sequence for these fragments, you're actually inferring the sequence of the entire 5 kilobases between it without doing that sequencing. Hence, you basically have much greater coverage, which is known as bridged coverage. With 1/6 to 1/10 of a lane on Illumina, it depends if the next generation machine, it'll be 1/50th of a lane, one can generate 7x coverage of a genome. And with this, one can then characterize a genome, whereas it takes 100 gigabases of sequence to completely sequence a human genome with whole genome sequencing. With 5 gigabases of sequencing, one can cover the entire genome with MP-Seq. This does not produce the sequence of every nucleotide, but still characterizes the genome, characterizes alterations. Now, at the Mayo Clinic, this has all been work that's been pioneered by Dr. George Vazmatzis, who's the head of the biomarker discovery program for our Center for Individualized Medicine. And working with Sarah Johnson, they've developed new algorithms to examine the data. They've also, because in some of the cancer things that we're doing, we're very interested in viruses and viral associations with cancer, so they concatenated all the viral genomes together to make a new artificial chromosome, which they call chromosome 29. They have powerful algorithms to detect novel junctions caused by inter- and intrachromosomal translocations, and there's a copy number variation tool that has also been added. And this is the result of one of these types of sequencing from the cancer that my lab studies, which is known as oropharyngeal squamous cell carcinoma. This is a cancer of the base of the tongue and tonsils, and it is increasingly being caused by human papillomavirus infection, and there's indeed an epidemic increase in the incidence of this cancer. It is estimated in the next couple of years the incidence of this cancer will be greater than the incidence of cervical cancer. And here is the sequencing and actually showing an HPV integration that has occurred into chromosome 15. The sequences at the top are all sequences onto chromosome 15. The sequences at the bottom have all been into the HPV-16 genome. Hence, this is an HPV-16 integration to the human genome and is a human viral junction that has been caused. And the little white thing on the bottom is actually showing the copy number of the HPV virus, which is considerably greater than HPV just being integrated. Hence, we can see that there's not only HPV integration in this cancer, but still a high episomal copy number of HPV present. Here is a second different integration, and this is integration into, I think, chromosome 2, and one can see the integration. All the things at the top are DNA sequences from chromosome 2. Everything on the bottom is to another HPV-16 genome, and you can see this integration. And here, too, there's a high copy number of HPV, so HPV is not only integrated, but is still present in episomal copies. And with this, one can characterize the physical status of a virus and a cancer. But in addition, mate-pair sequencing characterizes changes across the genome. Now, in addition to whole genome sequencing and mate-pair sequencing, there are also the sequencing of small gene panels, and this is perhaps the most popular use of next-generation sequencing in the clinic today. The strengths of this include focus on the genes of interest, hence the overall cost is considerably less. And this is the strategy that is being employed by the Mayo Clinic, but also by many other places as well, especially when one is worried about reimbursement for the clinical test that you are performing. Now, in order to capture either a small gene panel or even the exome, one needs to do some type of enrichment for the specific fragments. One very viable one, and actually there are a number commercially available, is SureSelect, which is the technology that is currently employed by Agilent. And one utilizes basically biotinylated baits for the sequences that you want to pull down. You take the genomic DNA on the top left, fragment it, and put PCR primers on the ends of it so that when you purified hybridized fragments, you can PCR amplify them. So one has these biotinylated DNA baits that is mixed together with the whole genome, and you can purify, it's basically fishing, you can purify the fragments you want. You then purify those out with streptavidin-coated magnetic beads which bind to the biotinylated baits. You then pull those out, PCR amplify, and the DNA fragments are then ready for sequencing. So the applications of clinical NGS, and I show something that's actually from hereditary colorectal cancer, where traditionally with Sanger sequencing, one might sequence, for example, the MLH1 gene. There are more than one gene that's involved in this, so one could do MLH1, MSH2, MSH6, and other things, and then add more and more genes. And once you get away from a single gene, you suddenly realize that the cost for doing it gene by gene with Sanger sequencing becomes very expensive. One might as well adopt next generation sequencing. Once you get over 5 genes, it's so much cheaper to do it that way. And then you have the flexibility of doing 5 genes, 10 genes, whole exome. This would be the 20,000 human genes, or ultimately whole genome sequencing. Now, whole exome sequencing is considerably cheaper and easier than whole genome sequencing. One has baits for 38.5 megabases of the genome, which is called the exome. There's also something known as the expanded exome, which has additional sequences in a couple of non-coding transcripts, and this is only 50 megabases. The advantage of this over whole genome sequencing, again, is that it's cheaper and there's much less stuff to analyze, much less stuff to store. And whole exome sequencing is currently being employed by Washington University instead of the small gene panels. And the realization is the overall cost is not much greater than a small gene panel. This provides comprehensive exome information, and it helps to build databases for the future, but there is the current issue of reimbursement for that, which is one of the reasons that a number of places, including the Mayo Clinic, are still focused on the smaller gene panels. RNA-seq is also a powerful technique to examine what is actively being transcribed. It provides information on message abundance, but also provides transcript isoforms and digital information on expression, and one can identify coding versus non-coding transcripts. In addition, with RNA-seq, one can detect fused transcripts, and indeed Mayo Clinic is currently exploring the utilization of RNA-seq for the examination of fused transcripts, which have a number of important clinical utilizations. Now, the clinical applications, currently Genomic Health for their assay to determine for basically the Oncotype DX utilizes real-time RT-PCR to measure gene expression for their assays, but would RNA-seq be a better alternative, more sensitive and more accurate? In addition, RNA-seq complements small gene panels, whole exome sequencing, and whole genome sequencing, because with, for example, whole genome or whole exome sequencing, one can tell mutations, but you don't know if that fragment that has that gene that has a mutation is actually transcribed or not. So RNA-seq complement these other types of things. And this thus concludes my presentation of the types of clinical applications of next-generation sequencing. And what's very exciting about this is how quickly this is being utilized in the clinic and will definitely be transforming clinical care. Thank you for your attention.
Thank you, Dr. Smith, for your informative presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Smith will answer as many questions as time permits. So without further ado, let's hop in and start asking a couple questions. Our first question of the day asks, why isn't whole genome sequencing utilized more routinely in clinical practice?
There are a number of reasons why whole genome sequencing is not used more routinely. In the same way that whole exome sequencing is still a little too expensive, and the issue about reimbursement, the total cost of whole genome sequencing is considerably greater than just the cost of the sequencing, since one has to assemble the sequence together, one has to annotate it, one has to interpret it, and more importantly, one has to store that sequence. Other limitations are the fact that when one does whole genome sequencing, really less than 2% of the genome is the coding portion of the genome. So what is the importance of characterizing changes outside of that important region of the genome? All of these things are limitations. This is the main reason why, with the notable exception of the neonatal intensive care units for diagnosing what is wrong with a newborn, we're not seeing much clinical adoption of whole genome sequencing yet. But indeed, with improvements of the overall cost of sequencing, I believe that we're going to start to see whole genome sequencing take off within the next few years.
Thank you for that answer. Our second question of the day asks, how can we get to the $100 genome and beyond?
That is an excellent question. With the NovaSeq 6000, we have the capability at the current cost for reagents when the 3 terabase flow cell is available, that the cost for the genome sequencing could be as low or less than $300. However, one needs further improvements. One has to bring the cost of the whole genome sequencing down to $100. My suggestion that is a company like Illumina decreases their reagent cost to bring it to $100, and then if the cost of the other components, for example, library preparation, analysis, and storage are decreased, one has the total cost is less than $1,000. However, even more improvements, if more improvements could be made, greater output so that sequencing could cost even less, then we could start to see an approach towards a $100 genome. My suggestion to all sequencing providers is that they should reach for that target, because when genome sequencing, analysis, and everything costs $100, we will have sequencing of almost every person on the planet, and that is the true revolution.
Thank you for that answer. Our next question asks, what are the limitations of MP-Seq?
That is an excellent question. While MP-Seq is a very strong viable alternative to whole genome sequencing, like whole genome sequencing, it requires DNA fragments that are of a sufficient, actually MP-Seq requires even larger DNA fragments. When one does MP-Seq, it is essential that one utilizes DNA which has not been stored in paraffin. So FFPE samples are not a viable solution. And this is a serious problem because paraffin characterization of samples is what is routinely done clinically. So MP-Seq will require a change in clinical practice away from formalin fixation. And indeed, if one wants to do any type of next generation sequencing of whole genome sequencing, for example, or MP-Seq, one is going to have to change clinical practice. And until that is done, this isn't going to be a viable platform.
Excellent. Thank you so much for that answer. It looks as though we have one more question. Our final question of the day asks, what about storage of data? What does that cost, and what are the potential solutions?
This is the true black box of whole genome sequencing. When one does a test at the Mayo Clinic, we have to store that data for 20 years. And I'm not even sure what is the total cost for storing a whole genome sequence for that much time. So we either need solutions where only a fraction of the data is stored, or, and this was originally made, and it's not a joke, the thought that one should do the sequencing and not store the data at all, but store the libraries, and then when one wants to do it again in the future, one can do it then. But this is an issue, and this is a serious problem. This is something that needs much more focus. It used to be that one of the key bottlenecks of whole next generation sequencing was data analysis, and while that still is somewhat of an issue, the question of data storage is a problem, and we need more viable solutions. I've heard of a number of places that are trying to figure ways to store a fraction of the data. You don't need the whole genome, you just need the alterations in the genome, and that is something that is currently being developed. But again, if we say, what is the cost for sequencing a genome, one has to factor everything in, and one cannot ignore the storage of that data, and we need some viable solutions for that before whole genome sequencing can be done routinely. Thank you.
I would like to once again thank Dr. Smith for his presentation today. Do you have any final comments you'd like to leave us with today?
Actually, I do. I just want to say that next generation sequencing represents a very important singularity. This is a term which is coined from Werner Vinge in science fiction, but is the technological development which completely transforms the world, and next generation sequencing is that singularity. I've had my own laboratory for 30 years. I am an expert on new and evolving technologies, but no technology that I've seen offers the capability of total transformation of both the research and the clinical space. So I advise everyone to become aware of this technology and to hold on to your seats, because this technology will transform everything. Thank you.
Thank you again, Dr. Smith. I'd like to thank the audience as well for joining us today, and thank Labroots for today's educational webcast. This webcast can be viewed on demand through May 2017. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. This is Christina from Labroots.com. We hope to see you here next time. Goodbye.